Artificial Intelligence Doctor. User question. What are the underlying mechanisms by which vinegar influences blood sugar regulation? GPT-01 answer. Vinegar, particularly its active component acetic acid, has been shown to influence blood sugar regulation through multiple physiological mechanisms. These mechanisms work synergistically to reduce postprandial, after eating, blood glucose levels and improve insulin sensitivity. Below are the key ways vinegar affects blood sugar regulation. Slowing gastric emptying. Mechanism. Vinegar delays the rate at which food leaves the stomach and enters the small intestine. Effect. This slower gastric emptying results in a more gradual absorption of glucose into the bloodstream, preventing sharp spikes in blood sugar levels after meals. Evidence. Studies have shown that acetic acid can prolong the gastric emptying rate, leading to reduced postprandial glycemia. Inhibiting disaccharidase activity. Mechanism. Acetic acid inhibits enzymes like sucrase, maltase, and lactase that break down complex carbohydrates into simple sugars. Effect. This inhibition slows down carbohydrate digestion and glucose absorption in the intestine. Evidence. Research indicates that vinegar can reduce the enzymatic activity responsible for carbohydrate digestion, leading to lower blood glucose levels. Enhancing insulin sensitivity. Mechanism. Vinegar improves the body's response to insulin by increasing the efficiency of insulin receptors. Effect. Enhanced insulin sensitivity facilitates better glucose uptake from the blood into cells, lowering blood glucose levels. Evidence. Clinical trials have demonstrated that vinegar ingestion improves insulin sensitivity in both healthy individuals and those with insulin resistance. Increasing peripheral glucose uptake. Mechanism. Vinegar activates certain cellular pathways, e.g., AMP-activated protein kinase or AMPK, that promote glucose uptake in muscle cells. Effect. Increased glucose uptake by peripheral tissues reduces circulating blood glucose levels. Evidence. Laboratory studies suggest that acetic acid stimulates glucose transporters, enhancing glucose uptake in skeletal muscles, promoting glycogen storage. Mechanism. Acetic acid enhances the activity of enzymes involved in glycogen synthesis. Effect. More glucose is stored as glycogen in the liver and muscles, decreasing blood glucose concentrations. Evidence. Animal studies have shown that acetic acid supplementation leads to increased glycogen storage in tissues, modulating incretin hormones. Mechanism. Vinegar influences hormones like glucagon-like peptide 1, GLP-1, that regulate insulin secretion. Effect. Enhanced GLP-1 levels stimulate insulin release, aiding in blood glucose regulation. Evidence. Some research indicates that vinegar intake can increase GLP-1 secretion, contributing to improved glycemic control. Reducing hepatic gluconeogenesis. Mechanism. Vinegar suppresses the liver's production of glucose from non-carbohydrate sources. Effect. Decreased endogenous glucose production by the liver lowers fasting blood glucose levels. Evidence. Studies have observed that acetic acid can inhibit key enzymes involved in gluconeogenesis. Summary. Vinegar influences blood sugar regulation through a combination of slowing gastric emptying, inhibiting carbohydrate-digesting enzymes, enhancing insulin sensitivity, increasing glucose uptake and glycogen storage, modulating hormone levels, and reducing hepatic glucose production. These mechanisms collectively contribute to better postprandial glycemic control and improved insulin function.